I will cover some sort of overview of the project and focus on the phenotypic traits, and then Romulo and Phoebe from Q will be taking on with the genomic analysis. We've tried to assess British silver birch and how well it grows in different environments in Britain from the genetic and environmental perspective. We had several aims, but within this talk, we will present 4 of them. We wanted to assess the performance of populations that were planted at 3 sites. We wanted to see and understand the genetic diversity of the silver birch, see whether phenotypic traits are actually heritable or can we predict them with genomic data, and finally identify genetic variants that are associated with environmental conditions. The map on the right shows the 29 provenances that were part of this study. They are represented by white dots, and you can see that they are quite well spread throughout the country. And then with the black dots, you can see the 3 trial sites. So there is one in Scotland near Loch Tay called the Drummond Hill. Then there is one in Norfolk near Thetford, and called Thetford, and one in south of Wales called Llandovery. They were planted in 2001, so these are adult mature stands. And we measured several characteristics, several phenotypic traits, some quite standard for forest research like DBH, and some quite novel like carbon discrimination. And we also collected samples for the DNA analysis. On top of that, we've collected samples from 157 plus trees of silver birch. So plus trees are trees that have some sort of favorable characteristics. They're usually quite straight. They're usually low in branching. They exhibit nice growth. And they are potential trees for any breeding program that we want to establish. So the meaning of this part of the project was to see whether we can find some sort of signal in the genome, whether these trees are actually being superior. So I think it's important to see the visual representation of the trial sites. You can see immediately that the sites differ from each other quite greatly. Especially Drummond Hill was on a wet and harsh site on a slight slope. So trees were basically throughout the years sliding down the hill and made some sort of banana shape growth, which was of course posed many challenges when we analyzed the data. Why do we want to work on provenance trials? Well, mainly because they're this amazing opportunity. They offer amazing opportunity to see the differences in different genetic material on site. So here you can definitely see the differences between the provenances that is planted on the left, which is quite thin, with the provenances planted on the right. So although they were planted on the same time and they have fairly similar growth conditions, they exhibit different growth patterns. The huge amount of time in the year 1 and part of the year 2 was spent on gathering all of the data, so on field work. Sometimes where we couldn't reach the fresh leaves for DNA extractions, we also used this quite finicky method to scrape the phyloderm below the bark, which is quite rich in good DNA. And then on the right, you can see how we measured resistance of the metal pin going throughout the trunk, which can be approximated then for wood density. And this method has been used for the first time in our department on such high number of trees. We've collected the following phenotypic traits. And we've collected many more actually, but quite some of them, especially historical data, we were not able to pinpoint them to individual trees. So these are the traits that we focus our first paper on. I wanted to show this picture just to say that projects like CFP are not beneficial just for these direct scientific outputs, which are, of course, papers for us, but they also produce many, let's say, byproducts. In this case, we've developed our own software for analyzing resistance data from the microdrill. And it was actually developed by the CFP intern. So it's another benefit of having internship programs as well. What I wanted to show is that even though you collect some, let's say, data that is quite clear, we are not always comparing the same thing. So here we see on the left quite thick tree and on the right quite thin tree, and they are roughly the same dBH as the next two trees. But here you can see that one tree is growing almost horizontally in Drummond Hill, yet still being alive. And some trees die throughout the project, and these are all things that you need to take into the account. When we've done some sort of first exploratory analysis, we saw that if we order the provenances from from south on the left to the north by latitude, we've seen some sort of declining trend in the traits. This is for the dBH, but when we plotted also other traits that we looked at, we saw the same sort of pattern. So trees that were growing in the south on average had some sort of better performance in terms of the dBH, in terms of wood density, in terms of carbon discrimination, in amount of carbon-13 isotope, which is some sort of proxy for water use efficiency. So they were more water use efficient. And finally, they were also taller. Not only that, there are several other aspects beyond the provenance that affect the growth. One of the very standard thing is the edge effect. So trees on the edge grow differently than trees within the stand. And using the models, you account for these differences, so your predictions are better. And finally, you want to look not only data from individual trees, but take into account some sort of broader population level. So just because trees are growing faster and seemingly better and they're healthier looking, it doesn't mean that they are more adapted, right? So here we see the mortality rates for different provenances growing in different seed zones. So seed zone 40, which had the highest mortality rate, is actually a seed zone that covers provenances from, let's say, South East England. And they had higher mortalities, especially when growing in the north. So even though they were looking healthier and they were taller, they also experienced higher mortality at the beginning of the experiment and throughout the experiment, actually. Once you take all of this into account, you can make multi-environmental models. And again, we've seen these clear differences, firstly, between the sites. So trees growing at different sites show different growth patterns. But also, again, this latitude effect of the provenances. So trees in the north, on average, had lower height, had lower dBH, but also were less water efficient, and they were having higher wood density. I will now ask the next speaker to take the control and continue the talk. Thank you.
Thank you, Domen. Yeah, my name is Romulo. I work at the Royal Botanic Gardens with Richard Buggs and also Phoebe, who's going to be speaking after me. And we were looking more at the genetic side in collaboration with FR, who did what Domin has just described. So one of the things we want to evaluate was the genetic diversity of the birch populations we had genetic data for. And when we summarize the variation at the genetic level, we have this PCA here. If the populations are extremely different from each other, you would expect clusters of populations spreading in the axis of the PCE. But what we see is that most of the provenances, they fall within this big cluster here, suggesting that there's not a lot of genetic differentiation between the provenances, even though they're across the country. There are 4 provenances, 2 in the north and 2 more towards the center of the country that are actually a bit different. But when we investigated what was going on with those 4 provenances, we found out that actually it's not that they are genetically very dissimilar from the others, it's just that there are a lot of related individuals in these provenances, so sometimes even siblings. So we think the PCA is capturing some sort of family structure within these provenances. And we also identified the plus trees, which are in bright red, and it turns out that they fall within this big cluster, suggesting they have a similar genetic background from the other provenance. So the main conclusion here is that there doesn't seem to be a lot of genetic differentiation between the provenances in the country. So as I mentioned, when we did a investigation of relatedness, looking at pair-ware relatedness between individuals within provenances, we found many first and second degree relatives. So this kinship plot here is just individuals against individuals and how related they are, and the more yellow, the more related. So you can see that in kind of Chase too, one of the provenances, you have a lot of clusters of yellow, which means that a lot of individuals are very related to each other. Whereas in Goldming, for example, you do not have that many. This seems to suggest that for some provenance, not all, the sample strategy was not as sufficient to get the whole of diversity of that population. So perhaps in the future, sampling more mother trees or mother trees that are more separated in space would be beneficial. One thing that we found was a bit we were not expecting is that some provenances had individuals that are related across provenances. So one individual be related to another individual in a different provenance, even though they were geographically quite far apart. So this could either mean that there's some, there were not many individuals, by the way, it was just, 5% of the trees had this kind of issue. And it was restricted to just a couple provenances. But that seems to suggest some sort of error during seed collection labeling, or it could just simply mean that there was some planting and both provenances used a common source, for example, plus trees. So we cannot separate these 2 possibilities. But it was interesting from a logistic point of view. And also we found a few tetraploids, suggesting there was some downy birches as well in the data, probably because they look so similar. But again, these are a very small proportion of the data set. And then we moved on to looking at the genetic basis for the traits that Domin was discussing. So we were trying to find out whether there are genetic variants that are associated with the trait. So here, for example, we have carbon discrimination as an example. So this is what we call a Manhattan plot. Is just simply plotting the p-values of all those associations. We have millions of variants in the genome, so you're looking at each individual variable and whether it associates with the phenotype. So whether you have a copy or 2 copies of that variant, whether you have, let's say, a larger DBH or not. So each dot in this plot is a variant. This is a p-value on your y-axis. It's logged and negative, so the higher the number, the lower the p-value, the stronger the association. And here the chromosome, where that variant is located in the chromosome. I ran 3 models, and when you have a large peak, that seems to suggest there is a region in the genome that is strongly associated with that phenotype. And one of the models gave a lot of peaks, as you can see, which would be very promising, very exciting. But it turns out that this model doesn't take into account the structure in the data. And in this case, structure simply means this latitudinal effect that Domen showed, because birch colonized the country from north to south. So there are a lot of variants that are present in the north that are not present in the south. So this ends up being picked up by the model. But when you control for that in the model, as covariates, for example, which are the other two models in the two different colors here, you can see that most of those peaks disappear. So it seems to suggest a lot of this variation that we're seeing here, they are just correlated with the phenotype, but they are not causally related. But still, when you control for that latitudinal effect, you still find 51 variants associated with some of the traits. This is not a lot. It seems to suggest that a lot of these traits are controlled by a lot of genes. They're polygenic, and the effects of each gene is very small, so it's not picked up as a significant effect in this GWAS. So why why did I choose to show you a model that it's imperfect? Because this peak here, when we started looking into it, we realized it was a very thick peak, suggesting it was a very big region of the genome, around 400,000 base pairs, that was, somewhat associated with the phenotype, but it could be just the noise from the structure. And when we looked deep into it, we found out that it's most likely a genomic inversion. So inversion is simply a bit of the genome that gets swapped over. So it's in a different orientation from the other individuals. So if you plot the variability in that inversion location, you see three clear clusters, which we think are the haplotypes. Red would be the individuals that do not have the inversion. Birch is diploid, it has two copies of each chromosome. So green would be individuals that have one copy of the inversion, and blue individuals that have both copies. Even though the blue one here looks a very small cluster, we have actually around 800 individuals clustered here. So this also seems to suggest that this inversion is quite recent, because there hasn't been a lot of time for variation to occur in this inversion. Otherwise, you would see something like this, something more spread. And as you can see here, although it's a bit small, you can see that this inversion follows a latitudinal climb, and that's why it was being picked up by that GWAS. So the inversion is much more, the frequency of individuals with two copies of the inversion, it's higher in the north. So when we do some analysis taking to the account, trying to control for this latitudinal effect, because remember, individuals in the north are smaller, for example, on average. But when you compare individuals within the same provenance that have or do not have the inversion, we do find an effect in the phenotypes, particularly DBH, water use efficiency, and wood density. So it seems that even taking the latitude into account, individuals that have the inversion tend to have lower values for these phenotypes or for these traits. So it's something that's still tentative, but it's interesting in itself. We don't know exactly. We looked at the genes in this region. None of them is extremely clear. There are a lot of them involved in apoptosis, cell death, perhaps root morphogenesis. So maybe there is a link there, something we should pursue in the future. And next, we look at the heritability of those traits, just how much of the variation in the trait is actually driven by genetic variation. So the higher the value in this column here, it means that more variation in the trait is explained by the genetics. And as you can see, some of the traits have quite high heritability. So height, for example, has 0.62, which means roughly that 62% of the variation in height is due to genetics. Same with wood density, but others not so much. Carbon use is different at 0.3. Steam form has very little genetic basis, but that seems to correlate well with what we found in domain analysis, that a lot of provenances which are genetically different were growing in different, achieving different heights no matter where they were planted. We also did a genomic prediction, so it's literally just a model, like you're attempting to learn the information from the genetics and trying to predict the phenotype of other individuals. So you try to see how the genetics relates to the phenotypes of a testing set, and then you select a different set training set, and then you use a test set to see whether the breeding value that you're estimating from the genetic data correlates well with the phenotypes of those individuals. And we can see that the accuracy of these predictions of the model are moderate. For height, for example, we have an accuracy of 0.58. And of course, when the heritability is low, the prediction is also very low. But these accuracies are, compared to the literature, are reasonably good. So that means that in the future, these models could be used in a breeding program for speeding the breeding cycle, because you don't have to wait for the trees to grow, as adults in order to predict how well they're going to perform. You can just look at their genetics from the model and see whether they have a high or low breeding value, right? And finally, we use those models that we trained with the provenance trees to estimate the breeding value of the plus trees. Because as Domen said, these trees were selected presumably because they have some merit with respect to their height or form. And what we find is that when we calculate the breeding value for those trees using the models, it turns out that for some traits, the breeding values for those trees are actually higher than for provenance trees are at similar latitudes. So if you look at this regression, you have latitude and the breeding values. The breeding values tend to decrease with latitude, which is, again, what Domen showed. Trees in the north tend to grow smaller, for example, no matter where they're planted. But if you look at, for example, at high latitudes here for height, the purple line is the plus trees. You can see that they have, on average, higher breeding values than trees from provenances at the same latitude, which seems to suggest that the selection for those plus trees was successful, that they are, if you were to plant these trees or offspring from these trees, they would tend to grow taller than picking individuals at random from similar populations. So now I pass the word to Phoebe.
Hi, I'm Phoebe. I'm just going to explain a little bit about the Genome Environment Association side of the project, which saw us associate the local climatic conditions of each of our silver birch provenances with their genomes to identify genetic variants that are exhibiting an association with ecological gradients. So we ran this analysis on 16 environmental variables, covering a range of classical temperature and precipitation climatologies, as well as wind and terrain. And through this analysis, we identified 218 genetic variants as associated with the environment. And as you can see from this bar chart, the majority of these were found linked to wind speed, but we also found variants associated with annual precipitation, water flow direction, mean temperature of the wettest and driest quarters, number of frost days, and TPI, which is topographic position index. And this actually covers quite a wide range of variables to which silver birch are having to respond to, highlighting these factors as key selection pressures requiring the greatest genetic adaptation. So as I mentioned previously, the highest number of associated variants were identified for wind speed, which in itself is quite interesting given that silver birch are a wind-pollinated species and their seeds are also dispersed by wind. Though of course wind can also be a stress for trees, it's responsible for uprooting and increasing rates of evapotranspiration. And upon closer inspection of the Manhattan plot for wind, which is very similar to the one that Romulo was showing before, so each dot is a variant, the genome is along the x-axis, and then the higher up the variant on the y-axis, the closer its link with the environment. And we found that the variants were within these quite defined regions of the genome, and these are known as peaks. And these areas then show a very strong link with wind speed. And we can take the highest variant from each of these peaks and we can plot its frequency within each of our populations against the wind speed measured at the local conditions of each of those populations. And in some cases, we find the genetic variant to show a really nice trend where the frequency of the variant decreases as wind speed increases or vice versa. But in other cases, this pattern was a lot less prevalent, with the one outlying provenance with this much higher occurrence of this variant, and this leads to a much more spurious association. And this outlying provenance was actually seen across many variants and environmental conditions. So we think that perhaps something strange is going on with this population, so maybe it was planted rather than wild, which is leading to quite different frequencies than other even neighbouring provenances. So one way that we can better characterise these associations is through annotating these variants and seeing which genes they are within or very close by. And by doing this, we identified genes such as endoglucanase and lacase, which are involved in cell wall strength and expansion, as well as phosphoglycerate mutase, previously shown to be involved in stomatal control, and aspartate aminotransferase, which is involved in energy production. So this just highlights these very specific genes as potentially involved in responding to wind, and therefore possible candidates for conferring greater environmental resilience. So in order to determine whether our silver birch provenances are locally adapted, we then looked at the suitability of each of our 29 provenances to the 3 trial sites that they were planted in. So we might hypothesize that provenances that are located closer to the trial sites in terms of geographical distance, but also ecologically, will show a greater suitability to that trial site if they are locally adapted. And these plots just show the differences in environmental conditions between each of our trial sites, which are shown by the triangles, and then each of our provenances shown by the purple dots. So these plots have just collapsed the variation across a whole range of environmental variables onto just 2 axes. And it becomes quite apparent that the conditions at Drummond differ quite considerably from each of our provenances. So it exists separate in the plotting space, and it's outside of this dark grey envelope. And Drummond is actually noted as being this particularly harsh site with a steep slope and quite great exposure to wind and temperature. And so this might explain some of the differences that are shown here. On the other hand, Thetford and Llandovery have local environmental conditions that are much more similar to each of our provenances, and they are within this provenance climate envelope. So we might expect that genetic suitability of each of our provenances to Drummond is a lot lower than for Thetford and Llandovery, because the provenances are locally adapted to a different set of ground conditions. So we then predicted the genetic suitability of our provenances to each of our trial sites by using the 218 genetic variants that the GEA had found associated with the environment. And we predicted their frequency in each of the trial site conditions. So we can then compare the baseline frequency of each variant in the original provenance location to the predicted frequency in the trial site conditions. And the difference between these 2 values then represents the amount of genetic change needed for that provenance to be best suited or adapted to that trial site. And this is often called the genomic offset or the allele shift, but it just represents genetic suitability. And we performed this calculation 3 times for each of our trial sites. And we found that the amount of genetic change needed for our provenances to be adapted to Drummond was a lot higher than that predicted for our more southerly trial sites, to which the provenances showed very similar levels of genetic suitability. And as I said before, we think this may be because the local conditions at Drummond are outside of this provenance climate envelope. But we do see that this provenance here, Elgin, has a very low predicted genetic change required to be suited to the conditions at Drummond. And Elgin is actually located in Scotland, quite close to Drummond. And so this lower predicted genetic change and greater suitability may be because Elgin is more locally adapted to this more exposed and hostile climate. Another very obvious outlier is Prestine, with this very high predicted genetic change for Llandovery and Thetford. And this provenance is located within central Wales. It's fairly close to Llandovery, and itself, it's not an outlying provenance from a genetic point of view. So we're currently still trying to look into this to understand why this is occurring. So with the wealth of phenotypic data that was also measured over the course of the trials, we then compared each of our genetic suitability estimates to the mean performance of each provenance within each trial site. So we might hypothesize that a greater predicted genetic suitability would result in better tree performance. If an individual is locally adapted, it should know how to grow best. And this pattern was very marginally seen in Llandovery and Thetford, where there is a very slight negative trend with greater predicted genetic change seen at lower phenotypic trait values. So shorter, thinner trees with thin bark were seen when the genetic change required was largest. But this pattern was not significant. For Drummond, this pattern wasn't really seen, and DBH actually shows a positive pattern with greater genetic change seen for trees that are actually growing the widest. But as we've already established, Drummond is outside of this provenance climate envelope, so this might explain some of these more contrasting patterns given these greater ecological differences. But this has quite practical applications for the use of genomic forecasting techniques that are used to predict genetic suitability, because they don't seem to work as well when the climates being compared are considerably different. So here, although we're predicting that a larger genetic change is required for the trees to grow at Drummond, the trees planted there are surviving, and although shorter, actually have a larger DBH than trees in other trial sites. And whilst it could be hypothesized that this is due to different phenotypes providing a selective advantage in different environmental conditions, it's previously been suggested that environments that are outside of the species range, the genomic forecasting techniques are less applicable for the species that are outside of the range. So they need to be quite carefully considered when you're predicting suitability to current or future climates. So just to quickly wrap everything up, we found very little genetic variation between our silver birch populations, but the southern provenances were found to have higher performance across all three trial sites in terms of height, DBH, wood density, and crown density. Our GWAS analysis found that phenotypic traits were largely polygenic, with many variants contributing a small but cumulative effect to the overall phenotype. Our genomic prediction models were able to identify the preferential growth of the plus trees, validating this approach. We identified local adaptation to wind speed, precipitation, and frost, and genetic suitability to the northern trial site was predicted to be worse, but the trees are still surviving there and are still performing well. So finally, we would just like to thank everybody at Forest Research and Kew who has contributed towards this project. We'd like to thank the CFP for funding, and thank you for listening.
Thanks very much, Phoebe. So we have some questions. So if I can ask our three speakers to put their cameras on. So the first question is from Annika Perry, who said, really interesting presentations. For Drummond, could the birch provenance that grew tallest but also had the highest mortality be demonstrating the risk of a grow tall fast strategy? Or do you think there's something else going on with these trees?
So, of course, we don't have like data to prove anything with this question, but I could speculate that this is what we had in mind. Yeah, so that's sort of provenances from the south basically continue growing, are just not used to the harsh conditions of the north. And there is one very interesting aspect, though, where we found out that trees of the northern provenances tend to close their stomatas later, meaning that actually they have higher, let's say, growth period than the southern provenances, but the south provenances still grow faster. And that's more of an interesting thing. But to answer your question with any sort of certainty, I sort of cannot.
Thank you. So the next question, this might be for you as well. So this is from Imam Syed. Does the material form the breeding population of the FTT qualified silver birch?
Yeah, so the plus trees that were analyzed are forming the FTT breeding population.
Okay, thank you. And do you know which populations are heavily presented in the orchard material, and are there any consequences?
Sorry, I do not. But if they are mostly from this FTT population, then we have nothing to worry about. They are either performing equally or slightly better.
Thanks. So the next one's probably for you, Phoebe, from Peter Jackson. What are the implications of the work on selecting the provenance of birch to cope with climate change?
I guess it depends on where you're going to plant them and what you're predicting your climate change to be, because there's lots of the like more practical, more like classical applications of the genomic offset work are to predict suitability to future climates. But I guess that because we're sort of ground truthing those sort of experiments where they're predicting this worst or best suitability to climates that are maybe not either going to happen or are too different, you might be over exaggerating to this like worst suitability, which isn't necessarily the case, as the trees in Drummond are still growing there and are growing, equally as well. And these phenotypes that we're seeing may be just adaptive to the harsher conditions at Drummond. So in terms of selecting provenances to cope with climate change, it depends where you're going to plant them and what you're predicting your climate change to be.
Yeah, so that would be perhaps some future analysis using future predicted climates, but using a similar methods to what you've done. Yeah.
I think
Phoebe.
ultimate. I think if you want to trust the genetic offset, then you just select those. You could run a model with just let's say the temperature variables and see which provenance would require a larger offset, and you would plant those that need a lower change. But I think our results show a more promising idea that, even the ones that require a big offset, they still can cope well with a harsh climate. So maybe you don't have to do anything.
Yeah. So there's actually lots of ability of those trees to adapt to the new climates anyway, with perhaps plastic responses. For you, Romulo, from Rachel Riley, regarding the geographically separated birch relatives, so where you've got those related trees from different provenances which are interpreted as anomalies, is it possible that they're not planted but are relicts of natural populations?
Well, the thing is they're quite geographically apart. So how could siblings or half sibs be present so far away from each other, or second degree relations? So I'm from the top of my head, it doesn't sound like a natural thing. It could be simply that, they were just planted from a common source, or even easier, because this provenance trials are such a logistic nightmare. There's so many things going on. So it's normal that within any project, you commit a small mistake and label one bag with the incorrect provenance. But it could be just planting. And it's very difficult to find out. You'd have to trace back, try to locate whether something went wrong. It's very difficult to differentiate what's going on. But the important thing is that shows the importance of genomic work, because if you hadn't done this genomic analysis, you would never know that these things may have happened. So I think this seems to suggest that for provenance trials, it would always be good to have some sort of genetic component to the project, so you can tell these situations.
Yeah, thank you. I don't know if you can answer this one because I don't think you had it in your data set, but Jonathan Burton Romulo is asking, did you find any influence of Betula, and I don't know if I'm going to pronounce this correctly, Celtiberica in the Welsh genomes?
For us to know where, let's say, we did find some triploids and tetraploids. They're not plotted in that PCA that I've shown. This was, it happened before I took over, so I don't have that data. But for you to be able to tell whether other species were contributing to the genome of the ones in your data set, you'd have to have some representative of those species. So you could plot them in a PCA, and depending where the hybrids would end up in the PCA, you could infer where the differentiation is coming from, where the introgression is coming from. So I'm not familiar with that species, but if we had genomes from that species and we plotted them together in the PCA with the hybrids, maybe we could tell whether some of the individuals had introgression from that species. But the answer to your question is no, we don't know. We know
thanks,
probably,
Romulo.
yeah, Betla, Downry birch, probably.
Could you explain a little bit more actually for everyone who's not involved in genomics when you're talking about the fact that you found tetraploids or other polyploids in the data set, what that means and how you know that that's a different species?
Well, it just means that some individuals have more copies of the chromosomes. In the case tetraploids, have 4 copies of each chromosome, and we know Betla pendula only has 2 copies. You can run some analysis specifically. It doesn't. We're not going to get into this, but you can infer how many chromosomes they have. And we know that Downry birch is a tetraploid, so it has 4 copies. So those tetraploids are probably Downry birch that were misidentified. And some of them were triploids, so that probably means that silver birch crossed with Downry birch and led to individuals with just 3 copies. So Downry birch dad offered 2 chromosomes, the mom silver birch gave 1 chromosome, and then they got mixed into it in 3. And it shows that even the triploids can survive, right? Because they were misidentified as silver birch and were planted and were growing fine. So yeah, it shows that hybridization, it's, if not adaptive, at least it's not damaging enough for the tree to survive.
Yeah, and once again, helpful to have the genomics work because that's difficult to pick up in any other way.
Yes.
Finally, a last question from Tim Gordon Roberts. I'm not sure which of you wants to pick this up, but what are the implications for, and we'll come on to this, Tim, in the next talk as well. What are the implications for policies that require planting local provenance?
Well, I think overall, it seems that the data is suggesting that it doesn't work for northern provinces, right? That you would, if you want, it depends what you want. If all you want is growth, if you're looking for timber, for example, all you want is a tree that grows very fast, then you wouldn't search for local provinces in the north. You would plant southern provinces, right? But then we would get into that discussion. Are the trees growing in the north smaller because that's an adaptive response for the harsh conditions, and that's just plasticity? Then in which case you could still plant local if all you want is just preserving the diversity of that region. But if it's for timber, then no, you would most likely go for southern provinces. Domen, do you?
Yeah, do you have anything to add to that?
Yes, I would agree that you can conservatively use some sort of rule of few, like 3 degrees from the south and plant it 3 degrees north. But when you have like a known harsh environment, I wouldn't gamble with survival and would plant something more local. So when you have like exposed areas, areas with late frost and stuff like this, I would definitely plant more adapted local material.